Hi, thank you very much for the kind invitation to talk about the impacts of new coronavirus variants. These are my disclosures. So what are we going to talk about today? I think you're all aware of the fact that viruses constantly change when they replicate. And this, of course, includes the SARS-CoV-2 virus. If you've been working in influenza, in HIV, you probably are very aware of this phenomenon because the SARS-CoV-2 virus has actually a lower mutation rate than these other viruses. So this replication leads to the occurrence of genetic variations over time and leads to the emergence of new variants. Some of these variants are not relevant and may disappear, but some might become relevant. And it is the changes of the genome that leads to relevant variants that we need to understand and learn about. And when I say become relevant, what I mean, they become relevant in clinical practice, they become relevant for the population because they might lead to the virus to spread more easily, they might cause more severe disease, or they may escape the body immune response. Hence, you can figure out this is really important. The cartoon in the slide shows the genome of the SARS-CoV-2 virus. And you can see that there are different areas of the genome. And over the next 15 minutes, we're going to focus mainly on the spike protein. The spike protein binds to human cells to allow virus entry. And it is also a common target for vaccine and therapeutics developments. So that's why it's quite important to learn about it. There are a lot of questions, very important questions that still need to be answered around variants. So do the variants lead to the spread of the virus more easily from person to person? Do they cause milder or more severe disease in people? Are we able to detect such variants by available diagnostic tests? And do the variants allow for the development of therapeutic that works against them so that we can have useful treatment for COVID-19? And also, do they lead to a change in effectiveness of COVID-19 vaccines? I don't think it's really clear today why these variants happen and what is the evolutionary selection of such variants. This all needs still to be characterized, but they seem to be the results of viral replication in people with chronic infections that have that are immunocompromised. So there is not enough immunological response to fight the virus that again continues to replicate for a long period of time. They may be the consequence of interspecies transmission. They may be the consequence and they may select because of therapeutic treatments like monoclonal antibodies or convalescent sera. They may be the consequence of limited cross-reactivity or of increased transmissibility. Now let's go back and think about the spike protein. So as you know, the SARS-CoV-2 virus is a single-stranded RNA envelope virus, and the spike protein is the major surface protein on the virus, and it plays a key role in binding to the host cell's receptors. It's the primary target for the host immune response against the SARS-CoV-2 infection and is an optimal immunologic target to use for the development of COVID-19 vaccinations. This slide shows the genome of the spike protein a bit more in detail. So there is a receptor binding domain, which is characterized by 222 amino acids between residues 319 and 541. And importantly, within the RBD, there is a receptor binding motif, which is really the bit of the protein that binds directly to the ACE2 receptor on the human cell, on the type 2 alveolar cell. And the receptor binding motif is also illustrated in yellow in the slide, where you can see some key positions, some key amino acids and areas where mutations may occur to generate the variants of interest, the variants of concern. Over time, especially from last autumn, you can see that the prevalence of the variants of concerns has been increasing. So what are these variants of concerns? Let's look at some of them. One is the UK mutant. The UK mutant is also called B117 and is characterized by certain key amino acid mutations and has been associated to an approximate 50% decrease in transmissibility rate. We will look at whether it is more severe, but also especially whether the vaccines that we have today are effective against this mutant. Another mutant is the South African mutant, which is also characterized by an approximate 50% increase in transmissibility rate. And again, it's characterized by certain key amino acid mutations, including the E484K, which is one of those mutations that really characterize variants of concerns because it seems to lead in associations with other mutations like the 501Y or the 614G, leads to a decrease in the efficacy of the immune response generated by vaccinations against COVID-19 that we have available today. And we're going to look at that in a minute. So another variant is the Brazilian mutant. It's also called P1, and it also is characterized by the 484K together with the 501Y and the 614G. So for those of you that haven't worked in HIV for 20 years like me, what are these numbers, just to make sure we're all on the same page, these key amino acid mutations? So we call them E484K. So it is the glutamic acid, which is indicated by the letter E, to lysine replacement, which is indicated by the letter K, at the spike position 484. So this slide shows the countries reporting the receptor binding motif mutants, and the UK mutant obviously is more prevalent in the United Kingdom, but you can see that it's also present in many other countries. Similarly, the Brazilian mutant was seen first in Brazil and is frequent in Brazil, but in many other countries, as well as the South African mutant that is present in South Africa and in many other countries. And then I have a slide dedicated to the Indian mutant itself. This is also called B1617, and it is characterized by the E484Q mutations at the level of the receptor binding motif, not the 484K. But you can see that the rate of identification of this mutant has increased remarkably over time, and this is probably the most talked variant in the UK at the moment as we speak. It's probably the most common topic of conversation in the UK at the moment. Now, let's just think for a moment on the potential change in effectiveness of COVID-19 vaccinations and the role that the variants may have. So I'm sure you're all aware that there is a vast array of vaccines under development. Some of them have already received preliminary approval, and you have received those vaccines probably. And the mechanism of action of the vaccines, the characteristics of the vaccines are different. We have mRNA vaccines, protein subunit vaccines, vector vaccines, and so on. I'm sure you're very much aware of that. And very briefly, because I'm sure you've been hearing about the immune response to COVID-19 vaccines, just to remind each other, the immune system responds to the antigens on the surface of the cell produced by the COVID-19 vaccine. The vaccines generate cellular immune responses, T-cell responses, and humoral responses, B-cell responses. And the immune responses include activation of cytotoxic CD8 cells that can destroy cells infected by the virus. They also lead to activation of CD4 T cells that increase both CD8 T cells and B cell responses. They lead to the generation of memory B and T cells that quickly respond to future viral infections. And very importantly, they activate B cells to produce antibodies against the SARS-CoV-2. So what happens when we look at the efficacy of convalescent or post-vaccination sera against certain variants of CoV-2 in vitro? You can see from the slide that the sera are effective against the wild-type virus and the UK variant, but the efficacy is decreased when they are incubated with the South African variant of the virus. And what about in vivo? What happens with what happens in people? So there are studies looking at vaccine sera neutralizations of the CoV-2 variants. Some are published, some are still on preprint. The majority of them are characterized by small sample sizes, but data are accumulating, so we're learning about this. 13 studies look at Pfizer, 3 look at the Moderna, 2 look at the AstraZeneca, 7 look at more than one vaccine, and 1 looks at an unspecified mRNA vaccine. 8 studies look at variants with one single or limited set of mutations. So this has a minimal impact because it is a combination of different mutations that would impact how much a variant actually alters the efficacy of a vaccine. The variants that have a larger effect in reducing the efficacy of vaccinations are those that include the E484K and the E484K combined with other mutations like the 417N or the 501Y. Importantly, is the South African variant that seems to lead to the biggest effect on vaccination efficacy. And the studies that included the South African variant showed a 3 to 11-fold reduction of efficacy of the vaccine, and this may also range up to 97-fold reduction. So in specific, let's have a look at some studies that look at Pfizer and AstraZeneca. The efficacy of the Pfizer vaccine following 2 doses is 93% against the UK variant, the B117, and it decreases to 88% against the Indian variant. And in terms of AstraZeneca, we're talking about a 66% efficacy against the UK variant and a 60% efficacy against the Indian variant. So at the moment, this is very good news for countries where the Indian variant is present and is frequent. But again, things change and are changing all the time. So let's have a look at the Novovax vaccine. This is a recombinant nanoparticle vaccines, including the spike glycoprotein and a matrix and adjuvant. they press released their data for the first time in January from the UK study and from the South African study. They showed a 96.4% efficacy against the prototype strain, the wild type strain, 86.3% efficacy against the B117, against the UK variant, but only 53% efficacy against the South African variant. So the good news is that it's still above 50%, but you can see that there is a remarkable decrease in terms of efficacy against the different variants. Now, finally, what about real-world data on vaccine breakthrough infection? So this is a study that was reported recently by the CDC in the United States. They looked at more than 10,000 vaccine breakthrough infection, and among these 10,000, 63% occurred in female, and the median age was 58 years. Interestingly, almost 30% of these were asymptomatic. 10% of patients with breakthrough infections after 2 doses of vaccinations were hospitalized, but 30% of these were asymptomatic or hospitalized for other reasons. 2% patients died, but 20% approximately of these 2% were patients who were asymptomatic or died for reasons that were not caused by COVID-19. So 5% of the reported cases and 64% of which of the 5% of the reported cases were COV2 variants of concerns. And this included the UK variants, the California variants, and the Brazilian variants and the South African variants. But you can see that the frequency of the Brazilian variant, the South African variants was still quite low. So in conclusion, I think we need more in vivo data on variants of concerns and breakthrough infections. These are emerging while the rollout of the vaccine is happening globally, especially in some countries where it's faster. Vaccines, however, can be modified to lead to immune responses against variants of concerns. This is great news. However, it takes time to modify the vaccines, and sometimes the variants may develop faster. And we saw that some of the variants lead to an increased rate of transmissibility. The development of the vaccines for coronavirus 2 has happened at a speed that has been unprecedented. So we need to trust the scientific and research community that this is possible, but obviously it's a big challenge. The role of variants may limit efficacy also of other therapeutic tools, such as antiviral drugs or monoclonal antibodies. This is something that we need to keep our eyes on, and it is a new area of the infectious diseases field that is developing. Importantly, the scientific community is focusing to target more conservative viral antigens and epitopes. So as we talked at the beginning, we saw that the target for the majority of vaccines is this polymorphic area of the spike proteins, these polymorphic genes that encode for a very particular and important area of the spike protein. And so again, the scientific community is looking for vaccinations or monoclonal antibodies targeting other areas, more conservative areas, where hopefully would not mutate and would not generate variants, and may allow for, again, vaccines and treatments to work for long periods of time. Thank you very much for your attention.